first major part of my career was very much focused on trying to develop new therapies for this advanced treatment-resistant stage of prostate cancer, and focusing very much on trying to understand and target androgen signaling in prostate cancer, because it seems to be the primary driver of the disease. But increasingly over time, we've been shifting our focus backwards, sort of trying to sort of see whether right at the time of diagnosis, can we predict the poor players and intervene there to try and reduce the emergence of this castration-resistant disease before the horse has bolted, and ultimately reduce deaths from lethal prostate cancer. Now, having said all that, you might wonder how we got into lipid metabolism. Well, the background was really around studying androgen signaling, as Lisa mentioned. And lipid metabolism is sort of an interesting area of research, because not only is altered metabolism a major hallmark of cancer, which I'm sure all the audience here is very cognizant of, it's also a highly androgen-dependent process. And so, because a picture, I think, speaks a thousand words, we've known for many, many years that when you incubate prostate cancer cells with androgens, we see a huge upregulation of lipid synthesis and other varying pathways in lipid metabolism. And this is a picture from our long-term collaborator, Johan Swinnen at KU Leuven. So we've known this for many years, and we now have a much better picture of the molecular mechanisms by which this occurs. And we sort of hypothesise that the reason that this is so strongly androgen regulated a process is that it does provide some sort of benefit to prostate cancer cells to enable them to grow and survive. But hopefully that might expose some novel vulnerabilities that we could target, perhaps with some of the less intense side effects compared with global androgen suppression, which is the standard of care today. So over the last almost 10 years now, we've been working as a consortium of both national and international researchers to better understand the role of lipid metabolism in prostate cancer. And for a long time, especially in the early days of research, it was very much focused on standard lab models of prostate cancer, such as cell lines and mouse models. We really wanted to take that back to play to our strengths, which was the fact that we have ongoing biobanking. We work a lot in tissue models from prostate cancer patients. So we wanted to really grasp the bull by the horns, and as difficult as it was, to try and understand and characterise the so-called lipidome of clinical prostate cancer. So using the resources of the Australian Prostate Cancer Bioresource, there's also a note of that here. It really started here. We have a long-term collection in Adelaide as well. We set out to look at blood, tumour, and also patient-derived models, such as the explants that my lab works on, to undergo a series of assays, including Johan Swinnen here, his work doing quantitative mass spectrometry-based lipidomics on the samples, but also combined with the spatial imaging of lipids that our centre in Adelaide is well known for, but is also complemented in KU Leuven as well. And sort of using these kind of techniques, we're able to get a pretty good picture about both the baseline prostate cancer lipidome, how it compares to the normal prostate, but also how it's dynamically altered by androgens, because that's really where our interest in this started. Now, we've published this work a couple of years ago now, so I won't go into all of the details of that. But when we first presented the prostate cancer lipidome, what we found is that there were some quite general, consistent changes in the transition from a benign prostate cell through to a malignant prostate cell. And unsurprisingly, that featured factors such as enhanced lipid synthesis, greater proportion of mono and polyunsaturated fatty acids. And so these were fairly well known from other models in the literature. A fairly new observation, which I'll get into a little bit later in the talk, was a preponderance of longer chain fatty acids in the lipids of prostate cancer cells. And these were things that we saw across many of the patients. But it's also true that by working with clinical samples, you also see a huge amount of heterogeneity in these sorts of specimens. And so this is just a circus plot showing at the individual lipid level just how much heterogeneity there is in terms of the changes that occur between matched normal and tumour samples from the same patients. So you can see that it's really quite considerably different between different patients. So, unsurprisingly, particularly when you go to the spatial level, you can see the explanation for this is just how even within a particular tissue or patient, there is so much heterogeneity within a tumour as well as between patients. So this is work that has been done and led by a couple of very good PhD students in my lab. So Shadrach Matuku, who is now a postdoc at Wollongong with Shane Ellis, and Jacob Trung, who's finishing up his PhD in my lab and is going to be working with Johan Swinnen in his lab next. But essentially, when you think about mass spec imaging, for those who aren't familiar with it, it's essentially like doing a mass spec in a grid-like pattern across a tissue section. So each little pixel is actually like a mini mass spectrum that then gets integrated together into, XY coordinates. So, it shares a lot of similarities with spatial transcriptomics in the sense that, the relative abundance across a tissue section can be inferred as difference in quantity of different types of lipids. And so what we see is that when, we have these very detailed annotated pathology done on these tissue sections, and what you find is that, they're often a complex mixture of just shown here in the pink is the tumour cells, the yellow is the benign epithelium, and then this kind of pinky blue area is the stroma. And so normally when we're doing that quantitative lipidomics that you saw in that circus plot, you're really getting an integrated mix or a smoothie almost of all of that together. So it's actually very difficult to pick out what's the source of any of the lipids that you're detecting. And so that's where spatial lipidomics comes in very, very handy, because what you can see is that when we do it, this is essentially a spatial heat map of areas of similar lipid composition, is that when you do that and you stratify them, the main differences are between these three key cell types. So the blue here, the stroma, the benign epithelium, and the tumour. And so because of this, you can actually zone in on these areas and really look very specifically at the lipid composition just in a tumour area, or just in a stromal area, or even that wasn't the case in this tumour, but if there's an immune infiltrate, you can sort of look at those kind of features as well. So that's actually been incredibly helpful. So this is, I guess, a nice illustration of the types of things that we do with this. So again, starting from our annotated H&E section, our first clustering based on lipid composition tends to split into epithelia versus stroma. The next split in the clusters is benign from cancer. And then you can actually go in and look at the abundance of specific lipid species. So some, for example, here are completely cancer localized, others are more benign localized versus stroma, for example. So by honing in on those, we can sort of get a better picture of exactly, what's going on, what's truly tumor derived or other components of the tumour microenvironment. And that's been enormously helpful in helping us understand how much is the microenvironment and how much is the tumour and how do they interact. So increasingly over time, my student Jacob has become interested in benefiting from one of the advantages that we have in our APCB biobank, and that is what we have now, very good long-term clinical follow-up on these patients. So many of them now have undergone either a clinical relapse or death from prostate cancer, which means that we can look back in time, essentially, from the time that they were diagnosed and see whether there were any particular changes in their metabolism that might have predicted whether they went on to have a very good outcome in the case of these patients here on the left, or a particularly unexpectedly poor or aggressive outcome. And the ones with no relapse, these were had to have at least 10 years of clinical follow-up with no evidence of relapse. So what we did is he actually imaged this cohort and was able to pick out the epithelium and stroma localized lipids, splitting that into normal and tumour as well. And to our surprise, he was actually able to find a number of lipids that did appear to be associated with patient outcome. So this is an example of two different patients here. One experienced clinical relapse, one didn't. And when we went through that mass spec imaging process that I mentioned before, we found that there were certain lipids that were present in epithelia regardless of the outcome of the patient. And here's an example of this that was present in both of these samples. But there were certainly other lipids that seemed to be specifically present in either the clinical relapse patients or in the non-relapse patients. Now, obviously, I've only shown you a patient of each here. But for these particular lipids, when we look across bigger groups of patients, we're seeing that there's this consistency in the changes, particularly for the cancer epithelium in patients that have gone on to have clinical relapse. So this has been, really exciting for us because we really weren't sure whether those changes in metabolism were happening very early in disease or whether it was an adaptive change that occurred as patients became resistant to androgen deprivation therapies. But it seems, all of these patients had not had any androgen deprivation at this point, so they're completely hormone naive. So this is something that's happening right at the start of the cancer journey. Now, at this stage, these sorts of markers are more exactly what I've just said, markers. How clinically relevant are those going to be? And the focus with that is really trying to identify what are the drivers of those lipid profiles. So what are the lipids? That's the number one challenge. So some of them are very easy to identify, some of them are more challenging. But if we can then relate that with other omics that we can layer on top in a spatial fashion as well, we can start to identify some of the drivers that might be targetable potentially. So I've just put as an example here where we've got our lipid signals here, we can have parallel slides where we can do spatial transcriptomics, for example, and this is something that we're doing quite actively at the moment. But I also wanted to highlight some other work that Jacob did to adapt the workflow with Brooker, who was the instrument company, to be able to simultaneously do lipid imaging and very detailed laser capture microdissection proteomics on these samples. So using those specific membrane slides, which enabled us to do it both on the same slide and actually let the lipid profile dictate where the microdissection was occurring. And using the approach that he optimized, we're able to get thousands of proteins from each cell type in the tissue, which was really quite remarkable. And so here's a nice example of where some of these relationships that we found really made sense. So, for example, we were able to show here, you can sort of see how detailed this microdissection is here. When we worked on these particular lipid profiles here, we're able to show that, for example, the increased expression of LPCAT1 that we detected in cancerous epithelium versus benign lined up with the expected changes in the lipid products of that enzyme. So, so this is an approach that we want to take forward into that outcome cohort to see whether the lipid profiles that do seem to be unique to poor outcome are being driven by an enzyme that we could then manipulate. So in summary, I've been able to show you that spatial imaging is very useful in many ways because it does give you that full picture of the heterogeneity of the tumour microenvironment, and it does, while we'll always need bulk lipidomics as well to help with identification of lipids. It's more quantitative, you can be more assured about the targeting, but it does give you just a huge amount of extra information about these very multifocal tumours, and coupling them with other omics is hopefully going to help us identify those mediators of altered metabolism. And the hope is that we might be able to better prognosticate the poor players in clinical prostate cancer, but link it to something that's then targetable, which is the ultimate goal. Now, what I've shown you so far has been a snapshot in time. So a tissue taken at the time of surgery, and then, you know, we watch what happens, but we've really still only got that one snapshot in time. We don't have follow-up samples from these sort of patients. It's not something that's very easily achievable in prostate cancer to obtain metastatic deposits. But what we do have the opportunity to do still in a tissue context is look at dynamic changes in lipid metabolism. And for that, I think many of you would probably be familiar with some of the work that we do in terms of patient-derived explants. So these are essentially pieces of tissue that we collect as part of our biobanking, but we take them back and culture them in the laboratory ex vivo. So we can culture those with various drugs and look for responses, and that's traditionally why we developed the model, was actually to evaluate new therapeutics in prostate cancer. But this sort of methodology of growing these tissues on a scaffold means that they retain their hormone responsiveness. So it allows us to actually look at hormone action in a whole tissue context as opposed to only the tumour cells themselves. Again, using lipid imaging, it's actually very handy because we can image drugs in tissues as well as lipids and other metabolites. So, for example, if we're trying to block androgen signaling using the clinical anti-androgen enzalutamide, we can actually image that in the tissue and we can actually line that up to various histological and target features in the tissue. And when we do RNA sequencing, for example, we can see that the expected changes occur in terms of blocking androgen signaling, but also many aspects of metabolism, as we might expect. Our lipid imaging has been very helpful in the sense that, again, as you would do in any other type of experiment, you can look in each cell population, whether it's epithelium, stroma, or others, and you can see specific lipid species or masses that are altered in response to blocking of androgens in these tissues. And so further analysis of this has really led us down a path where we've been able to identify a number of metabolic pathways and enzymes that we perhaps might not have considered otherwise. And so, for example, these are just some of the studies we've published over the last couple of years identifying various enzymes involved in fatty acid synthesis, metabolism, and in this case, elongation, that have led us to pursue these as novel targets. And so for the last few minutes of the talk, I just wanted to talk about some progress we've made with one of these targets, which is an elongase called ELOVL5. Now, fatty acid elongation is an absolutely critical part of fatty acid synthesis in any cell, and it's a very, at its very simplistic level, if you think back to your biochemistry days, it really involves the sequential addition of 2 carbon units to fatty acid chains. And this is catalyzed by these enzymes, at least in part, called ELOVLs. And elongation was one of the things I alluded to earlier when we first did our study of the lipidome of clinical prostate cancer. The one thing that absolutely hit us in the eye every time was that all of the fatty acid chains in the cellular lipids were longer. So there were very few that were shorter. They were really rare. Most of the time, all the fatty acids across the classes were longer. And, this was a bit surprising at the time, but now we know that this is actually a feature of lots of different cancers. So this has been published in other cancers, but also is something that seems to be dynamically regulated. So we've found that in acquired resistance of prostate cancer cells to numerous cancer therapies, we see elongation of fatty acids as well. In our explants, when we use the enzalutamide, we saw this huge shortening of the fatty acid chain. So we were sort of reversing that effect with anti-androgen drugs. So this was telling us that this is a very consistent effect and might be really important for cancer progression because, why would a cell use such an energy and carbon expensive way of perpetuating itself? It wouldn't make sense energetically unless it was really important. Now, again, since this has been recently published as well, I won't go into all of the details other than just to tell you that there are 7 currently known human ELOVL enzymes, but only one of those, ELOVL5, is expressed to any appreciable level in clinical prostate cancer, so either in primary or metastatic disease. And we showed in this paper that, in fact, it was a targetable enzyme in the sense that if we knocked it down, we could actually suppress prostate cancer growth and metastasis. And the one thing I will just highlight is that when we target it, it doesn't currently have an inhibitor, but either using indirectly through inhibition of ACC1 or through knockdown of a level 5 in those prostate patient-derived explants, we were able to suppress proliferation quite dramatically. Particularly, I'll show you here, even with a relatively modest, approximately 50% knockdown of a level 5 in the tissues, we saw a really dramatic reduction in the proliferation of those tissues. And that's really been driving us now to develop selective level inhibitors, which is another challenge in and of itself. So something new for our group, but is quite challenging. But just to finish, we wanted to understand if with that view to translation, if we're developing inhibitors, if we think this is a really important driving pathway that we want to try and target, we need to understand a little bit more about why it's being upregulated, why is elongation being promoted in cancer cells, what's the benefit to the cell? And so this is work mainly driven by Julia and Deanna, two terrific PhD students in the lab. They developed a series of a level 5 overexpressing cells, and what they were able to show was that when you overexpress a level 5 in prostate cancer cells, you do get elongation of those fatty acids in the cellular lipids. And this confers a growth advantage to the cells. So they grow faster, they form colonies better, and they also migrate and invade a lot better as well. So what might be the explanation for this? So we did have some untargeted and some targeted hypotheses about what might be happening. One of the targeted hypotheses was the fact that if you change the length of the cellular lipid fatty acid chains globally, you are going to have a pretty profound effect on the mechanics of the membranes within the cell. And so, for example, one of those that's been quite well studied are the membranes of the endoplasmic reticulum, or ER, such that when you have lipid elongation, you not only have a thicker, but you also have a less well-ordered membrane, and that can cause a lot of ER stress. And there's been a number of studies published recently showing that this alone can actually be an inducer of the unfolded protein response and a survival mechanism for cancer cells. So this is something we wanted to look into. Now, I won't go into detail here. I'm sure all of you are very familiar with the unfolded protein response as a cellular stress response. It consists of these three individual pathways and is generally studied in the context of proteotoxic stress, so an abundance of unfolded proteins. But obviously, we wanted to look in the context of lipid bilayer stress. So what we found is that when we profiled our level 5 overexpressing cells, we found that the unfolded protein response was one of the main upregulated pathways in the cells. When we looked at that at the individual gene level, what we found was that a level 5 overexpression induced genes from all three of the UPR pathways, so it wasn't one in particular. And we also confirmed that at the protein level shown here. Now, when we looked in more detail at the cells, we were able to notice that the ER didn't look normal. It was forming these whorl-like structures, and that's very consistent with ER stress. And we found that this was actually occurred in some quite aggressive clinical tumours as well. When we did some ER-specific lipidomics, so enriching for ER and then doing lipidomics on that, we confirmed that indeed our level 5 overexpression was increasing the length of fatty acid chains in the ER, and that was causing a resistance to agents such as tunicamycin, which are normally ER stress inducers. Interestingly, when we used inhibitors of each of the arms of the UPR, we were able to suppress that growth advantage that occurred when we overexpressed a level 5 in the prostate cancer cell. So it did appear that a big part of what gave those cells a survival benefit related to the UPR. And conversely, that when we targeted a level 5 by knocking it down, we also were able to reduce the expression of many of these factors in multiple different cell lines relating to the UPR and further enhancing response to tunicomycin when targeting. So our last question that we asked then was whether a level 5 and this UPR induced by this particular enzyme related at all to the very well characterised relationship between androgens and the UPR. So this has been shown for many years by multiple different groups that androgens do induce the UPR, and that had been linked in the past to transcriptional activation of many of the UPR pathway members by the AR. And indeed, we were able to confirm this in our hands as well, that UPR proteins and RNA is induced by androgens in prostate cancer cells. But fascinatingly, we found that this was markedly affected when we blocked a level 5. So for example, if you look here we have SciCon, we have knockdown of a level 5 in the absence and presence of androgens. And so we see induction of a level 5 by androgens as we expect, as well as the induction of these UPR proteins. But what we found is that not only did knockdown of a level 5 reduce the expression of these particular pathway members, we were able to almost completely block that induction by knockdown of a level 5. So all of this induction of the UPR proteins was blocked when we lacked a level 5. So this made us wonder, well, how important is this for LNCAP cell growth and viability? And probably our most surprising result was that when we stimulate cells to grow in the presence of androgens, we were able to completely block that induction by blocking a level 5. And not shown here, but we also found that that was the case if we block the UPR as well. So there seems to be this relationship between the UPR, a level 5, and the growth stimulatory effects of androgens, which I think is really fascinating. So we've done this in a number of different cell line models. We've also, because as I mentioned, we don't have a level 5 inhibitor, but we have some inhibitors of the level 5 pathway that we're able to use and showed very similar effects. So our next and final challenge, I think, is now going to be trying to understand the clinical relevance of this finding. So it's very exciting in these cell lines that, you know, the activity of one enzyme can actually block the effects of androgens. But is that meaningful in a, in a complex tumour microenvironment? So that's our next challenge to find out. So stay tuned. So to summarise the last part of the talk, I've shown you that a level 5 mediated fatty acid elongation is something that's very characteristic of clinical tumours. It's androgen regulated, and at least part of its mechanism is by induction of the UPR by inducing lipid bilayer stress in the ER. And I think what the main impact of this work to me anyway has really been changing my way of thinking about how androgens work, is that not everything is occurring through induction of various, big targeted programs of genes. In some cases, they're very bespoke changes to cellular membranes mediated by one or two enzymes that are actually causing this myriad of advantageous responses from the cancer cells. And so that gives us, I think, some really unique opportunities to think about how to target those most effectively. So with that, I'll leave it there. So thank you so much for having me. And I'm really looking forward to the rest of the symposium as well. So I'd like to just acknowledge the funding sources for our research. I've highlighted most of the researchers that were responsible for the work that I've presented today, but also our fantastic collaborators without whom we couldn't do it. And just as importantly, our research nurses, doctors, and the patients who donated tissues for this research. So thank you. Thank you, Lisa. That was a beautiful presentation. It's been great to see this progress over the years. So we've got time for one quick question. I'm going to keep a tight ship today, sorry. So would anyone in the audience have, oh, we do.
Jenny's the one.
It's lovely and a tough act to follow. I just wondered about a level 5 in other cancer types and whether we can get some hints about not just unfolded protein response, but also the ability of cells to move, etc.
Yeah. So it's interesting because each tissue type seems to have a different composition in terms of which levels it expresses, but there are a couple of other cancers that also seem to predominantly express a level 5, and one of those happens to be CML, so the leukemia. And fortunately, we happen to be co-located with a group that works on leukemia, so they're very interested in testing some of the inhibitors that we're developing as well, just to sort of see, is there any relationship in a blood cancer, which I think is kind of fascinating to me because you don't have that complication of the tumour microenvironment then. You really can look at the cancer in more of an isolated way. So that's one we're interested in following up.
Thank you so much, Lisa. So for everyone who's got burning questions for Lisa, I'm sure she'll be taking questions later in the lunch break. So our next speaker, very pleased to have our homegrown TRI, Dr. Jennifer Gunter. So she currently heads the Cancer Metabolism group at QUT at TRI. So welcome, Jenny.
Yeah, thank you. And I want to thank the organisers for bringing together the threads of cancer metabolism research from around Brisbane and beyond to host this symposium. It's been fantastic, and thank you very much for asking me to come along and give a talk today. I'll also be talking on prostate cancer. So we've had this beautiful introduction, probably saved me a couple of slides there, but hopefully demonstrating that prostate cancer is both a really fascinating cancer to study in terms of its metabolism, and hopefully we can exploit that. This is a bit of a viewer warning that this talk will contain slides with seahorse data, but actually just wanted to let this audience know that we have a seahorse extracellular flux analyzer here at the TRI. It's available for everyone to use. It's located on level 3. You can book training and the machine through PPMS, and I'm not a bad place to start if you want to start using that machine. But what I'll be talking about today is, I'm chuffed to say, a paper that was born from most of the PhD work of Charlie Bidgood, who many of you will be familiar with. And his paper was made available online overnight. Hooray! So just going to sort of work through this story, and we'll start with my own disclaimer that metabolism in cancer is very much more than just an enabling hallmark, and really underpins every single other cancer process. So there's my bias that I bring to the meeting this morning. Cells have an incredible amount of metabolic flexibility in order to service the needs at any given time as to what that cell requires. However, if we have genetic mutations or treatment-induced pressures, this can limit the metabolic choices that cancers have, and this creates a nutrient dependency that's not present in normal cells. And what we're hoping to do in the field is really exploit knowledge and try and apply that to improve diagnostics or prognostics, as you've just seen in the previous talk, or potentially therapeutics, and hoping that we can target these differences in the metabolic wiring and limit tumour growth while sparing normal cells. And this isn't just theoretical. There are a number of metabolic targeted drugs that are in advanced stages of trial, particularly in looking at metabolic dysfunction. So just really quickly, we've heard a little bit about what happens in prostate cancer, but normal prostate cells are highly oxidative, but they have a truncated TCA cycle. So truncated Krebs cycle, and this is because they've got a high expression of zinc transporters, which bring zinc into the cell and actually inhibit one of the enzymes that would convert citrate to isocitrate, and that citrate is actually exported as part of the normal function of the prostate cell. In the tumour, in the early stages of the tumour, we see that the zinc receptors are downregulated. There's not that inhibition. The TCA cycle somewhat is restored, but we also see that the citrate now is being converted or used in de novo lipogenesis. And we know that the lipids are very important to the prostate cancer cells. To really quickly recap, because I think you heard this in the previous talk as well, we treat prostate cancer. It's an AR-driven disease. We treat the disease primarily once it's spread beyond the primary tumour with a series of drugs that will either inhibit access of the androgens to the tumour through ADT or through more advanced and direct AR inhibitors such as enzalutamide. But this continuous AR inhibition drives major molecular reprogramming, and this includes changes to mitochondrial fuel pathways. And over the years, we've, from a large team grant that we had a couple of years ago, we've amassed hundreds of RNA sequencing samples that include cell lines, includes PDX tissue, includes patient-derived tissue that we've treated with and without these mechanisms of AR inhibition, either starving them of DHT or using drugs such as enzalutamide. And then we've tried to analyse that to see what are the really predictable changes that are leading to the failure of those, the eventual failure of those therapies. And we've taken a particular look at the metabolic pathways. And what emerged from that was an increase in a pathway which is labelled branched chain amino acid degradation. So branched chain amino acids include leucine, isoleucine and valine, and these are essential amino acids. Now the three of them share the same transporters for their uptake and have a number of shared enzymes at the beginning of their catabolic pathways. And these have been shown to be increased across numerous cancers, including breast, lung and pancreatic. We don't know much about branched chain amino acid degradation in the context, or we didn't, in the context of prostate cancer or many other cancers, but we know that it's critical in adipocytes. It both contributes to the TCA cycle for ATP generation, but it's also a really important fuel source for de novo lipogenesis. Our problem really has been that we've grouped these branched chain amino acids together when we do interrogation of large data sets like RNA sequencing, for example. They come up together in the same signature, really underappreciated or recognised that they've got quite differential and largely unique metabolisms of each of those individual branched chain amino acids. So I'm just going to briefly explain. So with leucine and isoleucine, these can both contribute acetyl-CoA, which would be contributing citrate to the TCA cycle and largely activating complex 1. Do you remember this one? Complex 1 in the electron transport chain, whereas isoleucine and valine would be contributing propanol-CoA, which can be converted to succinyl-CoA and would contribute succinate to the TCA cycle, and this would activate complex 2. Complex 1 is a mitochondrial encoded gene. Complex 2 is encoded by the nuclear genome. So we wanted to understand, well, what was the differential if we took away one or all of these branched chain amino acids, and what we noted was that our total lipid content dropped with the loss of one or more of these branched chain amino acids, indicating that they are indeed contributing to the lipid pool inside these cells. When we fed the cells, did the same experiment, but now fed the cells with a labelled amino acid, so this is C16 Bo-Dipi, we could see that, and now we included a cell line that was a non-malignant cell line, BPH1. We can see in the non-malignant cells, we had the largest response with an uptake of lipids, so trying to recover, we think, from a loss of lipid generation through the branched chain amino acids. That we saw the biggest effect in the non-malignant cells with leucine. the loss of leucine, whereas in our malignant cell lines, this is the LNCAP data, but we've looked in other cell lines, this was to valine. So we are seeing this switch from a dependency on leucine to valine, and this was blocked by inhibitors of, by 2-bromopalmate. So we think this is transporter mediated. So around the same time that we were fiddling around with these branched chain amino acid assays, there was a paper that was published which had taken patient-matched benign and malignant tissue and had undertaken some really exquisite respiration studies on these tumour patches, tissue patches, and had discovered that there was an increase in succinate respiration in the tissue that was coming from the malignant parts of the prostate. This is from prostatectomy tissue. And a reduction in the dependency on complex 1. So really seeing this complex 1 to complex 2 increased succinate respiration. And this is also seen in a later paper where this switch was associated with increasing Gleason grade. So this is a degree of severity within the primary tissue. So it began to seem like, well, maybe it's the succinate, maybe succinate dehydrogenase, which is the enzyme that would be using this succinate. Maybe that's probably what we want to be targeting, but we know from tumours, rarer tumours where we have mutations in succinate dehydrogenase subunits, that an accumulation of succinate actually acts as in and of itself and what they call an oncometabolite, so is able to drive oncogenic pathways just through its own accumulation. And this is at least in part due to the stabilisation of HIF-1 alpha and activating pathways downstream of that. So we probably don't want to be targeting SDH. And what we had thought when we put the work together with what we'd observed with valine was possibly, instead of inhibiting, we could potentially try and inhibit SDH activity. Not accumulating SDH might be to starve it of the fuel that it uses to create that succinate inside the cell, and potentially doing that via valine. How relevant is this outside of our cell lines then? So we actually approached the authors of this paper and asked if we could interrogate their data to have a look at what was happening upstream of the production of succinate. And Charlie derived this MITOS signature, which is the enzymes that get us from valine to succinate, and put that alongside the data. Now, this is the SLRF is the more red the boxes, the more reliant they are on the complex 2 versus complex 1 respiration. And we can see that the MITOS score and the ARPC score is also the androgen receptor and androgen receptor regulated gene signature. And we see a huge overlap between the MITOS score in these patients versus against the ARPC. So we're seeing an increase in the MITOS score from benign to malignant, and this correlates to their androgen receptor activity. We also put this in a data set by Lebrecq, who has subtyped metastatic CRPC samples based on their level of AR expression, whether they're AR high, AR low, whether they exhibit neuroendocrine features, or whether they're double negative. And we see a really strong correlation here with AR high expressing tumours, and we see actually a really low correlation with neuroendocrine in neuroendocrine tissue with, yeah. So let's pick an enzyme from this pathway. It was far more planned than that, but we picked on this enzyme HIBCH. And when we knock, so this would only be used by valine. When we knock this down in benign, our benign cell line, we don't really see any growth effects. However, in both androgen sensitive and castrate resistant cells, we see after about 80 hours, the growth of these cells is affected. And that correlates to an increase in the percentage of PI positive cells. So we're seeing an increase in cell death there. What probably surprised us was when we looked at the enzyme expression downstream from HBIC leading to the succinyl-CoA, so the enzymes in this pathway. When we knock down HBIC, we also see a reduction of these enzymes, and in fact, we also see a reduction in, spot the typo, in the subunits of SDH. So the knockdown of HBIC is really affecting then the expression of the enzymes in this entire pathway. So we were really wanting to link the importance of valine to these observations that had been made by the Scops group in this succinate dependency. And what we could see through high content imaging of the mitochondria is that when we knock down HBIC, we get this massive fission event, which is indicative of energy stress. We see a reduction in the mitochondrial membrane potential. And really the definitive piece of data that we needed to show was that through metabolomics, indeed, when we knock HBIC down, we do see that we are reducing significantly the metabolites of succinyl-CoA, succinate, fumarate, etc. What we weren't expecting was to see such an effect in citrate, and I'll come back to that in a moment. The seahorse data shows that we get, we were, I guess, anticipating that we would see a reduction in the amount of respiration from the mitochondria, which is shown here in the purple bars. This is in LNCaps and now a LNCap-derived enzalutamide-resistant cell line MR49F. What we weren't really anticipating was that we were going to see a reduction in glycolysis. But we saw that across our models, and this is the extracellular acidification data, or what is indicative of the levels of lactate that would be being produced by these cells. So we just were not anticipating that a change in valine availability was going to have much of an effect on the cells' lactate levels. If anything, we might have anticipated that it would go up as the cells became more dependent on lactate. But indeed, when we looked at our metabolomics again, we have all the way down the glycolysis pathway a reduction in those intermediate metabolites as well. And we're really still trying to come to grips with how potentially HBIC might be regulating this pathway. We undertook RNA-seq again. We're heartened to see that one of the major pathways that is downregulated was indeed the branched chain amino acid pathway. One of the ones that is upregulated in response to this knockdown being the biosynthesis of fatty acids. And I think based on our results of losing valine access in these cells, we might not be surprised to see that the cells are really looking to maintain their lipids in whichever way they possibly can, and really highlighting the importance of lipids in prostate cancer biology. And then Charlie did this analysis which really overlaid the metabolomics data with our RNA-seq data, and did this sort of joint pathway analysis, and which really allowed us to see what were the most significantly different pathways across those two data sets. And here is glycolysis sitting right up there with what we had anticipated in terms of the TCA cycle. So in summary, we now understand that valine contributes to the lipid content in malignant prostate cancer models, and that if we inhibit one of the major enzymes that would be converting this branched chain amino acid to something that the TCA cycle can use, we see a reduction in intracellular succinate levels by 40%, and we see reduced expression of the enzymes that would be utilised in that pathway. We know that inhibiting HBIC perturbs cell growth, oxidative respiration, glycolytic function, and the mitochondrial morphology in multiple models of prostate cancer progression and enzalutamide resistance. And this has got us a little bit excited that potentially we have a neck on the pulpit. We have a metabolic vulnerability that potentially we can target in this AR-positive advanced stages of disease. So thinking that HBIC might be a reasonable target against succinate-linked TCA remodeling in both prostate cancer, but potentially in other cancers that are where we see this increased reliance on succinate respiration. What might be driving that is probably where our future thoughts are lying. So we've actually got back in contact with the authors of this paper and brought them on board to try and help us understand some of their observations and some of ours. So they had published in this original paper that they see an increase in mitochondrial mutation frequency in cancer versus benign tissue, and that these really were, there was a hotspot of mutations that occur in complex 1, seems to be unusually susceptible to those mutations developing in that region, and is this driving these succinate changes. And so what we would like to understand then, on top of this information, is could this be a biomarker that would tell us whether a person was dependent on HBIC and the conversion of valine? So can we potentially use mitochondrial DNA burden as a biomarker of HBIC sensitivity? And that is because we are also on the search for inhibitors. So you would have noticed that all of the data was using knockdown of various types, and now we want to try and find whether there's an inhibitor that we can try and test and develop. And so we're undertaking compound library and drug screening and have developed a pipeline, and we'll be testing that through both an in vitro HBIC activity assay, but also a bespoke assay that we've designed on the seahorse, which really allows us to differentially understand how much of the respiration is occurring through complex 1 and how much of the respiration is occurring through complex 2, which is going to be critical in understanding the mechanism of action for any compounds that we have come across. And we have a couple of hits in the bag already. So with that, I will just acknowledge Charlie and the team at APCRCQ and our collaborators.
Thank you, Jenny, and congratulations on that new publication. Jenny also runs the Seahorse facility, if you didn't notice that plug. So definitely hit her up if you want to do any Seahorse. So we've got time for one question for Jenny. Yes. JP, thank you.
Thank you. I find it really interesting that there's a disruption of the TCA cycle in your cancer cells, but the same thing happens in inflammatory macrophages. Macrophages are big components of the tumor microenvironment and contributed to it. I wonder to what extent that contributes as well to the progression of the tumor. Any idea?
I could not tell you about succinate respiration in the T cells, but yes, the tumor microenvironment is critical to the way that a cell will adapt its fuel use, and understanding what's happening in the tumor cell in situ is going to be incredibly important. I think one of our priority experiments really has to be at this stage knowing whether we will have a durable effect by targeting HBIC with cells around it that might be happy to supply it with other fuel sources. And so that's the next question we want to answer. Yeah, great question. Thank you.
Thank you so much. Thank you, Jenny. So our TRI Cancer Seminar Series is prided itself on a diversity of different backgrounds and career stages. And so we've got a few short talks coming up. And so I'd like to welcome Megan Crichton from QT. So she's actually an accredited dietitian working in the cancer palliative care space. So I'd like to welcome you. Thank you.
Thanks, Lisa, and thanks for having me, everyone. We are definitely changing pace, and we'll be talking about some of our clinical research that we're doing in our Cancer and Palliative Care Outcomes Centre, particularly with nutrition interventions for cancer-related symptoms. So just an acknowledgement and paying respect to the Berrell and Yugara people where a lot of our research was conducted at QT, and acknowledging our collaborators as well. So there's obviously a team of dietitians, but we also heavily rely on the expertise of pharmacists and people in biomed as well to help us really understand the mechanisms of action. So please, if any of these topics interest you or spark ideas, please do get in touch. We are always looking for new collaborators in a multidisciplinary space. The first symptom of interest for us is chemotherapy-induced nausea and vomiting because it is common. It affects 20 to 70% of patients despite routine antiemetic prescription and continual improvements in these drugs. Lower gastrointestinal symptoms are also common, and the third symptom that we are interested in is chemotherapy-induced peripheral neuropathy. So that can affect 60 to 80% of people having neurotoxic chemotherapy, and there isn't really any good treatment for it other than stopping chemo or reducing the chemotherapy dose. It also is long lasting, so up to 30% of patients can still have problems up to 2 years after stopping their treatment. These symptoms just mentioned are among some of the most distressing for patients. They increase fatigue, anxiety and depression, which have a negative influence on quality of life, their food intake, and then subsequent effects on malnutrition and people's willingness to commence as well as complete their cancer treatment, which ultimately affect survival. So firstly, nausea and vomiting is probably one of the biggest programs of work in my space. We love to do a systematic review to get things started. I've put the QR codes next to some of our papers if you want to know or read any more, but today I'm just going to really skim the surface and packing a lot into the talk. So we started with looking at some dietary strategies for nausea and vomiting, particularly during chemotherapy. Not a whole lot of strong evidence out there. The best evidence supports nutrition education and support from health professionals with nutrition background, but what exactly that advice and education contains, we're not quite certain. There's a little bit of evidence around some adequate energy and protein macronutrient intakes and Mediterranean diet concepts, but a bit more work needs to be done there. In saying that, we were able to influence the international MASK guidelines for preventing nausea and vomiting during cancer treatment. And for the first time ever, diet and dietitians were considered in these guidelines. So that was pretty cool. We also updated some other guidelines, giving some more vague recommendations, not really based on strong evidence, but on the principle of that they're likely to probably do no harm and people can give them a go. Things like having frequent meals and having something in the stomach to help regulate stomach activity that's dysregulated in nausea and vomiting. We also have done quite a bit of work in ginger. So one specific nutraceutical that wasn't covered in those previous reviews. And again, started with a systematic review that highlighted the lack of robust trials, had highlighted some positive effects as well. So we explored that further in a randomized controlled trial, comparing ginger root powder in standardized capsule form with placebo. And we got our patients to take 4 ginger root capsules a day for 5 days, starting on their first day of chemotherapy. We recruited 103 participants and right before COVID, and we found some clinically relevant findings favouring ginger for a suite of symptoms. And interestingly, some symptoms related to nausea and vomiting as well. So your fatigue and nutrition status and malnutrition. This work did not influence the international guidelines, so ginger is still not recommended, mainly due to the conflicting dosing regimens used in the clinical trials. So we're currently updating our previous systematic review to pull all the previous evidence to date by meta-analysis. It's not published yet, so this is hot off the press, but there's about 15 more studies, including our large trial, and it sort of supports the effects that we found in our clinical trial. So that's exciting, and this may be enough to influence guidelines, but we're still not sure what the optimal dosing regimen is. This is the dosing schedule we used, noting that patients had to take the capsules 4 times a day, given the short half-life of ginger. We also looked at some mechanisms of action for ginger more broadly outside of the cancer space as well. We did an umbrella review looking at all the health effects of ginger, and I have a very embarrassing diagram explaining some mechanisms of action. This is about as deep as I go into the mechanisms in my work. But basically, ginger is thought to act in a similar way to antiemetic medications. So it's been found to block serotonin receptors, muscarinic receptors, and histaminergic receptors, stopping that nausea and vomiting pathway. It also might act on secondary pathways by inhibiting the oxidative stress and stomach motility abnormalities that are triggered by chemotherapy as well. We did another trial exploring whether the gut microbiota might be a possible mechanism of action for ginger, and we plan to do this in a subset of our chemotherapy participants, but then realised this has never been done in humans, so we started with healthy participants. And it altered some aspects of gastrointestinal bacteria and improved some symptoms of indigestion, but you wouldn't really expect big effects in this population given that they're healthy. But it does sort of path the way for future metagenomics work and analysing bacteria metabolites like short-chain fatty acids as well. So some future directions. Dose-dependent response trial is really needed. We're trying to get some money to do that. We also have a trial starting in a couple of months looking at the effects of ginger on stomach motility. And we also hope to look at some symptom clusters, particularly fatigue, and in conjunction with inflammatory markers. So is the effect of that we found on fatigue, is that because it's a flow-on effect of controlled nausea and vomiting, or is ginger having a direct influence on it? We won't know until we do some more work. We also have a program of work on lower gastrointestinal symptoms. So Anne Zhang is doing her PhD in this space, and she'll be looking at dietary fibre in addition to probiotics for managing symptoms during cancer treatment. She'll start with she's almost finished her systematic review, and then that will be used to inform a clinical trial. In the chemotherapy-induced peripheral neuropathy space, we have sort of highlighted the lack of additive non-pharmacological interventions in this space, and we are currently conducting, it will probably be one of the largest clinical trials in this space, led by Patsy Yates. It's been going for about 7 years. It's looking at heat therapy with massage therapy using little massage balls. It's quite unique in that it's a smart trial, so it mimics clinical practice. So if people do respond to the initial treatment they got, they'll continue that. If they don't respond, they get re-randomised. That sort of outlines the convoluted, chaotic flow diagram of the study. So the data analysis currently is a nightmare, and we'll also look there at the genomic analyses for some bloods we've got in the freezer. Future areas would be good for looking at whether there's a relation with nutrient intake and nutrient deficiencies and nutrition status with CIPN, particularly perhaps B vitamins. We've done a little bit of scoping and systematic reviews on medicinal cannabis, noting there that it does increase appetite. So this could be a potential future avenue for trying to get people to eat more and improve their nutrition. Validating a nutrition impact symptom tool in the coming months. We also have some PhD projects that we're currently looking for students for malnutrition prevention in people with diabetes and cancer, predictive factors for malnutrition post-chemotherapy, as well as trying to identify some nutrition biochem and diet intake profiles for some of these nutrition impact symptoms during chemotherapy to sort of guide some cancer-specific nutrition guidelines. That's it. In a flash.
Thank you, Megan. That was a beautiful presentation. We've got time for one quick question. We do. For anyone in the audience, anyone not shy? I can see lots of students who have burning questions. Okay, I'm going to go for it. So in terms of like dietary intervention in cancer patients, clearly you've got a cohort of people that would be very interested in trying to help with their cancer, but you've got a cohort that are kind of like, this sucks, I have cancer. How do you manage that kind of uptake in terms of your trials, and do you have a lot of people that actually just don't continue in the trial?
Yeah, it's actually really interesting because you would think that, but I think these symptoms are so at the forefront and so distressing to people that with our ginger trial, for example, 90% of the people that I approached participated in the trial because they're sort of desperate to try anything. And that's day one of their first ever chemotherapy treatment. They're willing to give it a go. I think people are kind of interested in that quick fix add-on as well. Doing a whole diet overhaul might be a bit different. In our CIPN trial as well, we followed those for 6 months after the initial intervention, and the dropouts were actually quite low too. So they're another sort of desperate population.
Okay. Interesting. Well, keep up the great work, and thank you very much for your presentation. So our next speaker today is a PhD student here at TRI, QT. Henry Lamb. He's in his final year and he'll be talking about peptides to target melanoma. Thank you.
So yeah, as the talk suggests, I'll be talking about peptides today as a tool to target metabolism. So a bit of a different spin. So I'll quickly touch on two parts of what I've been looking at in my project. So one part looking at serine metabolism, so kind of the context and the why, but then the method or the how being peptide scaffolds, and in particular a set of peptide scaffolds that we're interested in. So serine metabolism, being a non-essential amino acid, doesn't seem quite important, but we have seen in the literature that there are quite numerous studies showing that it's linked to drug resistance, and that comes in part with cancer metabolism allowing the cells that have that inherent flexibility during times of stress or undergoing therapy. And what I'm interested in particular is a protein, the rate-limiting enzyme in the pathway called phosphoglycerate dehydrogenase. And the downstream effect of this protein being upregulated is that you get an increase in serine and glycine, but also that links into the folate metabolism pathway, sphingolipid biosynthesis, phosphatidylserine synthesis, but also for purine generation. And this has been reported to be upregulated in quite numerous cancers. In my project, I've been looking at small molecule therapy-induced drug-resistant melanoma, but also been reported to be upregulated in bladder cancer, endometrial cancer, and other cancers alike. So focusing in particular on phosphoglycerate dehydrogenase, it is a functional dimer as a protein, roughly 56 kilodaltons in size. And a paper came out about 3 years ago showing that in the dimerization site, there are key residues that ensure the stability of the protein. And what they found that by disrupting these key residues, that you can actually disrupt the activity. So in green, so here we've got the catalytic site, but the dimerization site is the site of focus. And what they've found as well is that most pharmaceutical companies and also previous researchers have been unable to generate a successful small molecule inhibitor of the protein. And part of this is a common theme in cancer metabolism where you're dealing, especially in the glycolytic pathway, where you're dealing with a high concentration of substrate. And a lot of these inhibitors are substrate competitive inhibitors. So we need to look at alternative strategies to target these proteins. And so what this paper found is that two particular amino acids I'll focus on, which is this E108 alanine scan and isoleucine 121. These two mutations abrogated the activity, but also on the right-hand side, you can see that it pushed most of the population from mainly dimer forming to more monomer forming. And as I said before, what we found in the group, but also what we've seen in literature, is that these small molecules are ineffective in targeting the protein. So peptides, and why do we look at peptides as benefit? So one of the issues with targeting protein-protein interactions is that small molecules typically are unable to disrupt such a large surface area. Another benefit is that you aren't dealing with substrate competition, as I highlighted before. Another benefit with peptides as well is we have the possibility to engineer them to be cell-specific, so we can design them to be targeting cancer cell membranes with certain lipid types, whereas with small molecules or antibodies, at times we can have non-specific binding to the cell membrane and cell uptake. And while peptides, we're starting to see now an advancement in a range of cell permeable peptides, there's still some issues with trying to get peptides inside the cell and in particular into the cytosol. However, there have been other strategies in different ways to overcome just using peptide scaffolds alone to try and target proteins within the cytosol. Other ways have been looking at, for example, peptide drug conjugates, which can overcome some of the inherent flaws with small molecules, but also other strategies have included looking at peptide-peptide conjugates, or, for example, peptide antibody conjugates. And while peptides probably don't get as big a name as, say, the small molecule market, as a matter of fact, about 5 of the top 10 selling drugs worldwide are peptide-based. And that's largely in part due to the advancement in medicinal chemistry over time, where we've been able to improve the stability and circulating half-life of these peptides. So in the project, I've been looking at targeting PHGDH, and probably one of the first steps we do when looking at trying to target a protein like this is just doing mimetics of the dimerization site. So what I've done is designed roughly 5 or 6 mimetics that target the key residues that was listed in the paper, some that have more than one residue being targeted. And so in purple and red here is that dimerization site in particular, which is essentially like 2 L brackets forming an X on each other. And so we're trying to design peptides to bind to and inhibit that interaction. So the first one I was looking at was the inclusion of that first E108, and most of these peptide structures that are listed here have been solved with AlphaFold, so we still need to follow up and do NMR. We've also included isoleucine 121, but not the G140. And the other peptides have included other just different amino acids. They all have different lengths, these peptide sequences. Now, on paper, it is quite, not easy, but it is quite manageable to try and design these sequences and synthesize these sequences, but sometimes they can be a real pain to purify, and this probably took a good while to actually do so. And that's in part because we're taking small chunks out of the protein, and nature's optimized this process. So whether it be the folding or you get different byproducts during the actual purification process, there have been some difficulties. So now on this part of the project, I'm towards the step of actually trying to test this in vitro in enzymatic assays. From here, I think potentially it has, a chance of being one of these peptide sequences will be a drug moving forward. No, but it's a good starting point because we can use this for different screening methods and different screening assays to run better informed libraries rather than going in totally blind. So the second part I'll talk about in my project is looking for peptide scaffolds that we can use to either conjugate other peptides, or if you want, you can conjugate RNA or other drug molecules. And the scaffold that I'm interested in my project is called cyclogomecin. It's isolated from Brazilian tarantula, roughly 18 amino acids. This peptide has been cyclized, and all that means is that it is more resistant to being degraded by enzymes in the stomach, but also lowers the binding energy, so it's more readily able to get inside cells. The analog I'm looking at in particular has this mutation where it becomes more positively charged. And here when I have the RRCGM6, all it means is that I've replaced 3 of the arginines for the D analog in the hope that they'll be less toxic to cells. And so what we find is that this substitution from the L to the D arginines actually does reduce the toxicity. So this has mainly been done in cell lines so far, but we find, for example, in Hackett, a keratinocyte cell line, a BRAF mutant melanoma cell line, and a drug-resistant analog that we have quite a large window in the profile. And this is good because we want to use this DCGM scaffold to actually deliver cargo inside the cell. We also looked at this in a monomer and spheroid model too. While we do get a drop-off in the DCGM6 IC50, this is also, again, not necessarily such a bad thing, but probably what we need to look at with the peptides is how far they penetrate. So maybe look at some MALDI imaging with the spheroids to see how far our peptide actually can get inside the core of the spheroid. And in order to get a better profile for the peptide, we also look at the hemolysis, so how toxic it is to red blood cells. And what we find is that there is a large window, again, between this L and DCGM6 analog. And to do so, we incubate 0.25% red blood cell concentration for an hour with our peptides, and then we quantify the amount of hemoglobin that's being released. So far, we've got a peptide scaffold that is not toxic to the melanoma cells or red blood cells and can get inside cells. And so, sorry, quickly, I just showed that this peptide is able to get inside cells. At 4 degrees, we lose a lot of that uptake. So it implies that most of this is through via endocytosis. So that's probably one thing to look on is, can we get to escape into the cytosol and investigate if it has a preferential route of uptake. And I'll skip through the last part, but we've also looked at blood-brain barrier translocation and integrity. And so what we found in an in vitro model that these peptides are able to get across a monolayer for blood-brain barrier, and it also doesn't affect the integrity of this BBB monolayer after 24 hours at 1 micromolar and 0.1 micromolar concentration. So in summary, first part I've talked about is serum metabolism. The PHEDH has no successful small molecule on the market. And so peptides can be an alternative to that. And by designing peptides to target the dimerization site, we could potentially successfully block this protein. And the next part of my project is looking at testing it in an enzymatic assay. And for the peptide scaffolds, we have an ideal peptide scaffold that isn't toxic to cells, can get inside cells. And the next part is looking at the pharmacokinetics and pharmacodynamics in mice. We do have bioactivity data, but PK and PD, where it's not conjugated, will give us a better insight. And just acknowledgement to the lab and also collaborators. Thank you.
Thanks so much, Henry. And someone who's also working on peptide drugs, it's great to see all these peptides coming out of your group. We've got time for one very quick question in the audience. No. I think you're off the hook. Thank you, Henry. Lots of backup data that we would have loved to have seen with a longer presentation.
Should have prepared that one first. Eh?
No, good. Thank you, Henry. Thanks so much. And so moving on, we have Naomi Berrell. So she's a research assistant at Queensland Spatial Biology Centre. So we had a bit of a lipid spatial kind of idea earlier from Professor Butler's talk, but we look forward to hearing about your work. Thank you, Naomi.
Thank you. Thank you for the invite. So today I'm going to be talking about one of our recent projects that's just been accepted, so hopefully published very soon, on head and neck cancer. We're kind of new to the metabolic space, but we're pretty excited to be here. So I'm excited to share some of that work with you. So head and neck cancer is the 7th most common cancer globally, and around 900,000 new diagnoses per year, 450,000 deaths. Common risk factors include alcohol use, smoking, and HPV virus. Upon initial diagnosis, there's around a 68% 5-year survival, but unfortunately 50% of these patients will go on to have metastatic or recurrent disease, where there's only a 20% 2-year survival. Traditionally, head and neck cancer is treated with chemotherapy, radiotherapy, and surgery, but lots of patients do go on to have these treatment-resistant cancers. So increasingly, we're seeing immunotherapy offered as a last-line defense if they're eligible. So eligibility criteria is usually based on PD-L1 expression of over 1%. And we see that in other cancer types that usually predicts immunotherapy response. But in head and neck cancer, we see that only 20% of patients respond. And while they have an amazing response, 80% of patients aren't responding. And so our goal is to identify novel biomarkers that might better predict people's response to immunotherapy, as well as finding other markers that might help us personalize treatment or ultimately improve patient outcome. So this is just a little biorender version of a tumor. And as we know that a pillar of cancer is this dysregulated metabolism. So you could have dysregulation in amino acid pathways, oxidative stress pathways, glycolysis, or even fatty acid, for instance. And what we're trying to do with our spatial work is identify where these are occurring within the tumor and how they're interacting with other cells in the tumor microenvironment. So for instance, this is just 2 cores from our tumor microarray. This one's from ovarian cancer, but just in the red cells, we have GLUT1 as our glycolysis marker and PNERF in green as the oxidative stress. So you can see here that they're quite unique tumors, but they were both given the same drug. So the goal, I guess, with all these clinical trials coming out with metabolic inhibitors in conjunction with standard treatments is maybe in the future we can try and personalize medicine so that we can combine a metabolic inhibitor that might match, I guess, the tumor profile and personalize medicine in that way. So for our study, we had a tumor microarray of 25 cores, and half of which received immunotherapy. So there were 12 patients in the cohort. And we ran 2 different spatial atlases on them. So we did a 53-plex protein analysis with about 10 metabolic markers. And to do this, we used the PhenoCycler fusion. So this is cyclic IF imaging, stitching them all together so that we can visualize them all on the same tissue. And to complement this, we use the geomics whole transcriptome analysis. So this is a regional-based approach where you select regions within your tumors and then further subdivide them. So we put PAN-CK and PAN-C negative as a tumor and stroma regions. So I just wanted to highlight the heterogeneity that we're seeing across patients with the same disease. So here we've got someone who had stable disease, partial response, and progressive disease. And you can see just from the protein work that they are unique. But we can also overlay some of that RNA work that we did. So looking at the cytokine activity, you can see that it does vary across patients, but also within a patient core. We can do this for all the pathways that we want. So for instance, humoral immune response, tryptophan metabolism, and this granulocyte macrophage colony stimulating factor. But this is just highlighting the heterogeneity that we see across patients and why there's a need to personalize medicine. So this is just another core from our tumor microarray. And I just wanted to highlight how we're using the proteomic work to map back on and maintain that spatial integrity. So we can map different things such as metabolic pathways, cell phenotyping in neighborhoods, just as an introduction, I suppose. When we looked at the tumor metabolism within the tumor of using the protein work, we see that those glycolysis pathways and oxidative stress pathways were upregulated in non-responsive patients. This is in line with a lot of literature at the moment, where we're seeing these pathways associated with immune evasion and treatment resistance. Interestingly, we saw this lactate pathway up in the responsive group. So we've used LDHA as the marker for this pathway. When we look at the RNA work, we're seeing that in responsive tumours, we get this upregulation of iron transporters, cytokine activity, and humoral immune response and complement activation. But within the non-responsive group, we get signalling pathways such as the epidermal growth receptor, upregulation of cholesterol biosynthesis, but most interestingly, I guess, is the glycolysis pathway was upregulated again. So we're seeing that in both the protein and the transcriptomic work. And we're also seeing that hypoxia pathway up in the non-responsive patients. And so while I've spoken a bit about the metabolic work, we've also conducted other analysis, such as like cell interaction and neighborhood analysis, but I don't really have time to get into that today. So do check that out when it's published soon. So what's next is we'd love to keep exploring this metabolic space, in particular those oxidative stress and glycolysis that we're seeing upregulated, so expanding into larger cohorts and new cohorts. The ultimate goal, I guess, is to translate some of these findings into the clinic. And I'd like to thank the lab who's helped me out tremendously on the project, as well as our funders and of course the patients who generously donated. Thank you.
I'm always amazed at how beautiful those spatial cores are. It's beautiful. Thank you very much for your talk, Naomi. We've got time for a quick question from anyone. Oh, Lisa?
That was lovely, thank you. I wondered, is the methodology that you've been using, is it amenable to more frozen specimens? I was just wondering whether you could start to, add an additional layer with a metabolomic spectral.
Yeah, so we're using FFPE generally. I think that you can do frozen, but for the protein work, at least, there's a reduced amount of antibodies that you can run on it. So I guess pros and cons there, but I'm super interested in the moldy sort of work. So interested to see where that leads and to chat further.
Yeah, no, I'd be happy to.
I think we'll just take one more question. We have one from, oh no, she's turned around. Oh, you're good. You're good. Okay. Thank you so much. Thank you, Naomi. And finally, we've got Dr. Prahlad Raninga. So he's recently joined TRI, working with Kunkana's group, and this is his first talk at TRI, so welcome. Thank you.
Thanks, Lisa, for an invitation here and wonderful opportunity to present our work here. As Lisa mentioned, we recently moved from QIMR to MATER in March, so we're still starting up. So this is some of the work which was ongoing for the last couple of years. This is mainly, we are sort of trying to target MYC and amplified high-grade serous ovarian cancer by using different targeted agents. And we are still sort of new in the field of cancer metabolism, sort of, I would probably say, at the interface of targeted cancer therapy and cancer metabolism. So as we all know that high-grade serous ovarian cancer is probably one of the deadliest gynecological cancers, and it is the 5th most common cause of cancer-related death in women. The median survival rate hasn't really changed over the period of a couple of decades, and it still remains less than 5%, for less than 5 years. And the frontline treatment options for these patients is neoadjuvant chemotherapy, platinum-based chemotherapy, and surgical removal of the tumors. And recently, PARP inhibitors have been approved as a maintenance therapy for platinum-sensitive high-grade serous ovarian cancer patients. However, the biggest clinical challenge in these patients is the development of chemoresistance. About 80% of patients, they do develop relapse disease within 2 years of diagnosis. And currently, there are no effective targeted treatments which have less toxicity compared to chemotherapy. So, excuse me. MYC is a proto-oncogene amplified in about 50% of all human cancers, and high-grade serous ovarian cancer also about 50 to 55% of these patients have MYC pathway activation. And MYC is a nasty oncogene. It has its finger in pretty much every single aspects of cancer, regulates genes involved in cell cycle, apoptosis, proliferation, and metabolism too. And high-grade serous cells have survival dependency on MYC. However, MYC to date, although it represents an attractive therapeutic target, several efforts have been made to target MYC by using small molecule inhibitors, but none of them had made to the clinical trials, or even if they were trialed, they didn't show any efficacy. So people are using alternate treatment strategy called synthetic lethality, where we identify the genes on which MYC-driven tumors have dependency for survival, or the pathways that are often deregulated in MYC-high tumors. When we did a GSE analysis by using TCGA dataset from the patients, we found that proteasome pathway is significantly enriched in MYC-amplified ovarian cancer patients. And this makes sense because high-grade serous ovarian cancer patients have they have very high levels of chromosome instability, which often leads to proteotoxic stress. To counteract such a high levels of proteotoxic stress, cancer cells often upregulate proteasome machinery to remove the proteotoxic stress. By keeping this in mind, in past years, FDA-approved proteasome inhibitors have been trialed on solid cancers, including ovarian cancers. However, they have failed to show any clinical efficacy. And this is probably because if we look at the proteasome structure, it has three catalytic sites, beta-1, beta-2, and beta-5. The FDA-approved proteasome inhibitors like bortezomib, carfilzomib blocks beta-1 and beta-1 activity, beta-5 activity only. But when beta-5 is blocked, beta-1 and beta-2 gets upregulated to compensate for the loss of beta-5. But luckily, we have another proteasome inhibitor, which is like a natural proteasome inhibitor. It can block all three catalytic sites at the same time, leading to irreversible inhibition of proteasome. So we looked at the IC50 of metasomib across the panel of ovarian cancer lines as well as patient-derived tumor cells. And we find that a very nice correlation with MYC pathway activation in these cells. So MYC-high cells have they're more sensitive to metasomib compared to MYC-low high-grade serous ovarian cancer cells. And there's a nice correlation between MYC expression and metasomib IC50. Then to understand the mechanism of action, we did whole like global transcriptome analysis, RNA sequencing in OCARH cells. And we found, this was very interesting results, that we found one of the top pathways that were downregulated belong to metabolic pathways, pyrimidine and purine metabolism pathways. And we also did proteomics and to see what is the, do we see the changes at protein levels too? And interestingly, even in proteomics data also, we found purine and pyrimidine metabolism pathways are significantly downregulated upon acute metasomib treatment in these cells. So the pyrimidine metabolism pathway has two arms, salvage pathway and de novo pathway. Cells are heavily relying on de novo pathway, where basically they use glutamine and by key rate-limiting enzymes, CAD, DHODH, and UMPS, they basically regulate each and multiple steps of this pathway, leading to pyrimidine nucleotides into the cells. So we thought, does metasomib inhibit any of these rate-limiting enzymes? And at mRNA levels, we see a significant downregulation of CAD, DHODH, and UMPS. We did not see any effect of metasomib on the salvage pathway, suggesting that metasomib is mainly targeting the de novo synthesis, de novo pyrimidine synthesis. And when we did functional metabolomics, we could see that a key metabolites belonging to the pyrimidine pathway were significantly reduced in the cells upon metasomib treatment, which tells us that metasomib is targeting pyrimidine pathway. To further confirm this, we did a rescue experiment where basically we pre-treated cells with a pool of pyrimidine nucleotides and purine nucleotides. for 3 hours and then challenged the cells with merisomib. And we could see that supplementation of cells with pyrimidine and purine nucleotides are able to rescue the cells from merisomib-induced apoptosis or cell death, which suggests that merisomib is likely to inhibit pyrimidine metabolism pathway, which is then causing cell death in MYC-driven high-grade serous cells. We further validated our findings in vivo. So we used one of a very highly aggressive ascites-derived PDX model of high-grade serous ovarian cancer. Basically, we engrafted luciferase-tagged PDX cells into the mice, NRG mice, and within a week, we could see the tumors was established. And then we treated mice for another 2 weeks for merisomib. And we see a dramatic anti-tumor response in this aggressive PDX model of HTSOCs. And when we did, we had few mice which we collected after 1 week of treatment to see whether merisomib is still inhibiting pyrimidine pathway in this PDX model in vivo. And we could see that the mRNA levels of CAD, DHODH, and UMPS were significantly downregulated in merisomib-treated tumor cells compared to the vehicle-treated tumors. And when we did metabolomics on these tumor cells, we could see that the metabolites belonging to the pyrimidine and interestingly purine as well, they were quite dramatically reduced after merisomib treatment compared to vehicle-treated tumors. So the next question was, what is the correlation of pyrimidine pathway in high-grade serous patients? So again, we used TCGA dataset, and we performed a single-cell gene set enrichment analysis on basically MYC-high patients versus MYC-low patients. And interestingly, we found that, and then we were correlating basically whole KEGG metabolic pathways with MYC gene signature. And we found a very significant correlation with pyrimidine and purine metabolism, plus 1-carbon and oxidative phosphorylation. So if you look at the correlation metrics, higher the correlation metrics, better there is a positive correlation. And probably we saw the highest correlation with pyrimidine metabolism, meaning that MYC high-grade serous ovarian cancer patients have enriched pyrimidine metabolism. Compared to many other metabolic pathways. And we further did some clinical analysis to see what is the effect of pyrimidine on the survival outcomes in these patients. And we could see that basically we looked at the MYC-high patients, and amongst the MYC-high patients, if we examined whether MYC-high plus pyrimidine-high patients versus MYC-high versus MYC-high and pyrimidine-low patients. And we could see that the patients with MYC-high and pyrimidine-high have relatively shorter disease-specific survival. And the expression of key rate-limiting enzymes of UMPS, CAD, and DHODH was significantly higher in high-risk patients compared to the low-risk patients. So this is where we are at the moment. And in summary, we could say merisomib has significant anti-cancer activity in MYC-driven HTSOCs in vitro and in vivo. And merisomib exerts this anti-cancer activity via pyrimidine nucleotide inhibition in these cells. And pyrimidine metabolism correlates with worst prognosis in HTSOC patients. And hence, drugs targeting pyrimidine metabolism are probably a key, promising therapeutic approaches for these patients. So our next step is to, and there are some clinical evidence that pyrimidine pathway metabolism has been shown to be involved in chemoresistance. So we're currently validating if merisomib induced pyrimidine metabolism can overcome chemoresistance in these patients. And also we will, in future, we will target DHODH specific inhibitors and see what is their response in these patients or this HTSOC tumors. And finally, would like to thank my supervisor, Kumkum Khanna, and the lab members and all the collaborators who have helped throughout the project and funding bodies. Thank you. Happy to take any questions.
Thank you, Prahlad. That was beautiful talk and great response in mice. Do we have time for one question? Yes. Yep, Jenny.
Wondering about the timing of your co-therapy and what your rationale is for that, for moving forward in that way.
So timing for?
The timing, sorry. The timing of either sequential or co-therapy, or what your thoughts are on that, whether you have to create an environment where that metabolic inhibitor is going to be most effective.
Yes, so when we plan to put a combination therapy with platinum drugs, we'll probably do a sequential treatment and then hit the mice with merisomib first, just to take out the pyrimidine metabolism, and then maybe a week later. So we'll give 2 doses of merisomib and then challenge the mice with chemotherapy, just to create that environment of pyrimidine depletion.
Beautiful talk. Thank you so much. So thank everyone for their great presentations today. Thank you, Lisa, for making it from Adelaide. Your talk was brilliant. And thank you for everyone across Queensland who's joined. I'd like to let everyone know that lunch will be available in the atrium, but I also want to highlight the next instalment in our TRI cancer series, which will be an imaging-based mini symposium. So if you haven't already registered, please do so. We look forward to seeing you there. And I also thank the speakers. And if you could come to the stage and we'll have a quick photo, that would be great. Thank you so much.
So we have everybody down here. Can I ask you please?
Well, it's not, so I think it's not too French. Yeah. And we actually used to do these every fortnight, very much. Wow, you got reported this